I'd like to thank the organizers for giving me the opportunity to present on behalf of the Pediatric Task Force. One of the goals of the task force is to facilitate the genomic interpretation of pediatric tumors. And of course, we're well aware of some of the challenges in pediatric cancer. We have a very large number, diverse diagnoses. Some of these are very rare, they're emerging, they're not necessarily that well studied. Many of the drivers of these pediatric cancers are just not seen at all in the common adult cancers. Many of the alterations are fusions, which are notoriously more difficult to identify and to curate. And in some cases, the driver of a pediatric cancer may be in a gene we don't really think of as a cancer gene, and it may not even be included in panels. So what we've done is to generate a master list of genomic alterations that have clinical impact in pediatric cancers. And what we've done as a starting point is to go through the pediatric volume of the WHO classification of tumors and extract out all the genomic alterations that are diagnostic, prognostic, therapeutic, would present hereditary risk if in the germline. And we added a fifth category of drivers that may not meet the other criteria. And then from this master list, we have prioritized getting these into the public knowledge-based civic that you heard about yesterday that is freely available, no strings attached to anyone. And we have prioritized getting the defining diagnostic variants in there first. For those that may not be as familiar with the WHO classification, these are the 14 volumes, and historically they have been divided out based on tissue type and organ system. In 2022, the WHO released this first pediatric tumor volume. In the center are the chapters. You can drill down, and in this case, you see the renal tumors that are most important in pediatric patients. If you click on an individual diagnosis, in this case clear cell sarcoma of the kidney, you get to a very structured entry. And basically the hereditary risk variants typically are in the epidemiology section. The diagnostic variants are in the essential and desirable diagnostic criteria, and genomic alterations that have prognostic significance. And some of the ones that have therapeutic significance will be in that prognosis and prediction section. The WHO is quite good for hereditary risk diagnostic and prognostic. We do pull in information from the FDA and the NCCN guidelines for therapeutic. So this is just a snapshot of what this master list looks like. Each line is going to be a variant with clinical impact. Everything is tied to the ICDO diagnoses. This table is all gene alterations. So it's a fusion, there will frequently be a second gene. We indicate if we expect it to be a gain of function, loss of function, or gain of alternative function. And to help in pipelines, we indicate whether you would be looking for a short nucleotide variant, a copy number alteration, or a larger structural rearrangement, either intragenic like an ITD or the more common fusion or rearrangement. We give a rough idea of what the frequency is in the tumor type, and then we'll indicate whether it has diagnostic, prognostic, therapeutic, or hereditary risk implications. For diagnostic and prognostic, we are focused on the highest tier 1A, and that information, of course, is coming from the WHO. With therapeutic, we have included 1A, 1B, as well as what we call 2C with clinical evidence, because it can be quite helpful to have that information when the targeted therapies do work in these very rare cancer types, which typically aren't included in clinical trials. So overall, from the 277 non-benign pediatric tumors in the volume, about 2/3 had at least one genomic variant with clinical impact. And at the bottom, I just broke out how they separate in CNS tumors, extracranial solid tumors, and heme malignancies. And I'd like to draw your attention to the very high number of diagnostic variants that are tier 1A across the board. And this is quite different than what you would see in the common adult cancers. I also like to draw your attention, while fusions aren't the major alteration, you do have a significant number of fusions that are playing important roles in all of these. So we did think that it was most important to get the diagnostic variants that are defining for these very rare cancer types into the public domain, and so we're starting with what we call the defining Tier 1A diagnostic variants. So everyone is familiar with ASCO-CAP guidelines. It just needs to be listed as being diagnostic for the tumor type. The WHO will put the given variant either into essential or desirable criteria. And as we were going through, we realized that we needed additional classification, and we came up with defining versus supporting. So a defining variant is one that is only found in that one diagnosis, maybe one or two others. For example, if you have an EWSR fly fusion and you don't know anything else about the tumor, you do have a pretty good idea that you have a Ewing sarcoma. And same thing with a MyoD1 L112R mutation, you have a pretty good idea that's going to be rhabdomyosarcoma. Supporting variants aid in diagnosis when coupled with additional information. So for example, if you have an infant with a tumor in the lung region and you identify a DICER1, gain of alternative function, loss of function, you have a pretty good idea that's a pleural pulmonary blastoma. Same thing with SMARCB1 loss and rhabdoid and beta-catenin activating mutations in desmoid. So I know this is small, but this just gives you an overview of the variants that we designated as defining diagnostic variants in CNS, solid, and heme. And what we're doing with these for each one is to go through and generate 3 evidence items from 3 primary papers linking the given alteration to the diagnosis in CIVIC, and from that we'll generate an assertion. So if we look at the first one, diffuse astrocytoma, MIB or MIB-L1 altered, Minji is one of our pediatric neuro experts. Over on the left are the 3 separate evidence items where she curated the details of the different types of MIB-L1 alterations tied to this cancer type, and then we'll generate an assertion. And the assertion will have a general statement about the cancer type, the specific alterations that are seen, the role in the cancer, as well as the language from the WHO that makes this a Tier 1A diagnostic variant. This just gives an overview. You can see we're nearly done with the CNS. We've made significant inroads for the solid tumors, and right now we're focusing a lot of effort on the heme. And that's just with the purple and the green dots. And I'll tell you that our next prioritization, there are variants that are considered essential for diagnosis, but we put them in the supporting category. These are actually a lot more straightforward to curate, but what you do see is there are a lot of pediatric cancers driven by SMARCB1 loss, and in a number of cases, detecting SMARCB1 loss is considered diagnostic and essential diagnostic. We also have a number driven by beta-catenin. Over in CNS, you do have a number where you do need to identify an alteration in the RTK-RAS-MAP kinase pathway. So I'd like to thank the fantastic group that we're collaborating on this. You can see that we have good representation from a number of oncology centers across the US. And we have 2 fabulous contributors from the land down under, Diane and Lavinia. And the pediatric task force is headed by Gordana and Ashmoy, and Jason is our fabulous coordinator. If you have any questions, if you would like access to some of what I talked about before we get it all out there into the public domain, if you'd like to join the pediatric task force, please feel free to email me or Jason. Thank you.